My name is Marvin Warner, and I am a research scientist at Pacific Northwest National Laboratory. Currently, I'm researching the individual pieces of antibodies to set up a chemical reaction that will give off light just by mixing our reagents together with a sample that contains an explosive molecule. Basically, the immune system is designed such that when you have a foreign invader or something like a virus or a bacteria, it is designed to mobilize cells within a given body that are specifically designed to target the invader, render them basically inactive or neutralize them. This keeps any type of foreign body or invader from causing any adverse effects within the organism. You can exploit this immune system behavior to basically then produce antibodies that are specific for targets of interest. The explosive protein conjugate is injected into the organism. The immune system kicks in. Antibodies are produced against the foreign body in order to basically clear that foreign body from the organism. We isolate the cells that basically encode for those specific proteins that are designed to clear the foreign body from the organism. And then we take that genetic information and produce the proteins external of the organism. Basically, the development of the detection platform that we have in mind would have a great deal of effect for deployment at distant outposts or checkpoint type monitoring, where you would have very little to no external power. You could basically take a sample off of a surface, stick it into a handheld system or some type of container that contains your protein reagents. You would then press a button to release the reagents, and as the proteins come together in the presence of the explosive, you would see a light that would not otherwise be given off in the absence of the explosive. I think we're going to find better sensitivity and just better functionality overall by using a low-power detector compared to the conventional method of pulling a laser and a detector system and a computer and everything else into the field. You have a much smaller power footprint. You have the understanding of all the nuances and technologies that are necessary to form reagents that have been postulated in the literature and do so in a somewhat non-linear, non-conventional path. Can only happen with a team that you could form in a place like PNNL, where we have all of the different multidisciplinary components.